galaxy of intellectual guest professor who joined this webinar, and delegates, ladies and gentlemen. It is a matter of immense pride to stand here before all of you this morning. On behalf of SMBT Seva Bhavi Trust and entire organizing committee of ICTM 2021, we welcome you for second International Conference on Traditional Medicine 2021. It gives me privilege to invite Shri Ram Kure sir, Dr. Meenal Mohagaonkar ma'am, then Dr. Babar sir, Dr. Hire sir, Dr. Naik sir. Before we proceed further, I would like to felicitate all of them who honored the dais. First of all, I would like to request Dr. Yogesh Ushir, Program Chair, kindly felicitate Shri Ram Kure sir. Thank you, sir. Now, I request Dr. Ushir kindly felicitate Dr. Meenal Mohagaonkar, Dean of MBBS College. I request Dr. Ushir sir again to felicitate Dr. Pradeep Bhabad, Principal of SMBT Ayurveda College. Now I request Professor Kundan Tiwari to felicitate Dr. Hire sir. I request Kundan Tiwari to felicitate Dr. Naik sir. I welcome Dr. Chitare sir, Deputy Dean of ICMRC. I welcome you, sir, on behalf of SMBT Educational Trust and ICTM 2021 Organizing Committee. I request Dr. Ushir sir to felicitate Dr. Chitare sir. On the next, I request the program chair, Dr. Yogesh Ushir, kindly welcome all the guests and the delegates, speakers who joined this webinar.
Thank you so much, sir. So, good morning to all of you, respected dignitaries on the dais, national and international delegates. Namaste from India to my national and international delegates. I, Dr. Yogesh Ushir, warmly welcomes all of you for the second ICTM at the SMBT campus. I wish you best of luck for the grand session of this conference. Thank you.
Thank you so much, sir. Now, let me tell you about the theme of this conference. The second International Conference on Traditional Medicine. The theme is Development and Modern Trends in Traditional Medicine. Now, dear all, there is no specific treatment for COVID-19. Furthermore, people in the community and researchers are trying to find out the best way to cure or prevent this monster disease including herbal medicines. Since immune status of patient plays a vital role in COVID-19 infection, herbal medicine, which has an immunomodulatory effect, could have a potential as a preventive measure and even a therapeutic agent for patients with the COVID-19 infection. Ladies and gentlemen, with the thought of this, we try to put a focus on several traditional medicines which are currently preferred over allopathy. Considering the greater future perspective in the herbal medicine, we attempted to review the theme as a current status of its use in treating various diseases. Ladies and gentlemen, it's assurance from the entire organizing committee of ICTM 2021 and SMBT Educational Trust that at the end of conference, you will find yourself in a research at a level where you are expected to be. With this, I welcome everyone again, and I request all the dignitaries who honored the dais kindly raise for the curtain raising ceremony. Appreciate welcome everyone for this inaugural function of ICTM 2021 Second International Conference on Traditional Medicine. Thank you so much. I request everyone kindly occupy your seat. Now I request Dean of MBBS College, Dr. Meenal Mohagaonkar, kindly express your vision, your views regarding this ICT.
It gives me immense pleasure to present for the second international conference on traditional medicine organized by the Pharmacy College of SMBT. I congratulate the organizing team led by the Bashir and his all team members from the college. When I think of the Pharmacy College, I would like to say only a few words that in pediatrics, when a small child is born, we look after the milestones of the child. And there are certain babies who walk at a very early age. Some babies start walking as early as 10 months, start running at 11 months. And I think this holds true for our deep pharmacy college. At the institute, they have grown so fast, and it is the vision and dreams of Dr. Vishin and supported by all team members that they have achieved a lot at a very young age. I congratulate the team and my best wishes for successful conduct of this conference. Thank you.
Thank you so much, ma'am. These words are really mean a lot for us. Request the chief guest of this inaugural function, Dr. Sriram Kure, sir. But before that, I would like to introduce our chief guest. Actually, this is a profusely overjoyed to take this opportunity to introduce our chief guest of ICTM 2021, a person with a good heart, a person who always remains self-motivated and motivates others too, a well-known personality and a pillar of SMBT Seva Bhavi Educational Trust, none other than Sri Sriram Kure sir. He is the Chief Officer, Administrative Officer of SMBT Seva Bhavi Trust. I would like to request him to address the delegates.
Good morning, ladies and gentlemen. On this immense occasion of SPM inauguration, especially welcome to Dr. Mrs. Mohawk, Dean of the SMBT Institute of Medical Science and Research Center, and Dr. Sir, the Vice Principal, SMBT Institute of Medical Science and Research Center, Dr. Professor, Principal of the SMBT Ayurveda College and Hospital, Dr. Sachin Gile, the Administrative Officer of SMBT Seva Bhavi Trust, Dr. Nai, the Administrative Officer of SMBT Ayurveda College, Dr. Vishnu, the leader of this his colleagues, or the national and international dignitaries. It's a high time that such an event took place to highlight the remarkable contribution which traditional medicine made after so many years of our lives. A glance through the list of presentation planned for the next couple of days reveals the amazing diversity of this application. They range from the traditional medicine, their biological screening, quality assurance, and herbal drugs and their formulations. Traditional medicines have had a profound impact on the way we live. The SMBT ICTM is to have been a partner in the growth of these researchers in the field of healthcare. A keynote of ICTM is to provide a platform to the new ideas and research in the field of traditional medicine. It should contribute to the bright future of mankind. There is a great demand of traditional medicines. It has a great contribution in the healthcare sectors. Developing countries are not simply passive recipients of traditional medicine created and sharp by their developed partners. On the contrary, many developing countries have acquired high levels of research in the field of traditional medicine. There are great keynote speakers who are going to share their ideas and vision in this. SMBT Seva Bhavi Trust always encourages and supports events like ICTM, which contribute in the sector of healthcare. I am pleased and wish that researchers from the developing countries and well represented among the presenters this week. Ladies and gentlemen, the SMBT has a vision to make the SMBT Institute the most respected and respected members of the society. Respected and respected members of the institution in the field of education and healthcare training. Since 1984, SMBT is providing education in the field of pharmacy, Ayurveda, and allopathy. Afterwards, a multi-speciality charitable hospital was started by the SMBT Seva Trust with the sole aim of providing help to rural people in need of medical care. Over a period of time, the trust further expanded its charitable activities in the rural areas by starting various educational institutes. The SMBT is always striving for the best research activities and practices in the field of education and healthcare services. ICTM is another milestone towards this vision of SMBT Seva Trust to use its full potential for the benefit of mankind. Ladies and gentlemen, conferences such as this provides a valuable opportunity for research scientists, industry specialists, and decision makers to share their experiences. I'm grateful to the many experts who have come to share their knowledge this week. I also welcome the representatives of various educational institutions, national and international delegates, industry associations, and NGOs who have joined us. I'm sure you will have fruitful and rewarding exchanges in the next couple of days. I wish you every success with this important conference, and I look forward to learning about the outcome. Thank you. Thank you very much.
Thank you so much, sir, for sharing your deep knowledge, your vision, views towards IIT Educational Trust. Uh, Now, at the end of inaugural function, we are really grateful to Sri Sriram Kure sir, then Dr. Meenal Mohagaonkar ma'am, then Dr. Milind Chitare, Dr. Pradeep, sorry, Bhabat sir, then Naik sir, Hire sir, who came here for this inaugural function, who took out some time from their busy schedule. I hope you will find this conference highly engaging, fruitful, and beneficial as well. With the kind permission of dignitaries, I declare inaugural ceremony and thank you once again. Thank you so much. All participants and delegates who joined us recently, will be back to you and will continue the session in 5 minutes. Thank you.
पाठीमागचा बोर्ड आहे ना जरा इकडे घ्या
I hope I am audible to the delegates. Before I say something, before I introduce our today's speaker, I would like to tell delegates, if you have any doubt regarding a session, if you have any question, then kindly find the chat box and please put your query in that chat box. At the end of session, we'll back to the questions and we'll try to comply all the query that you put in that chat box. Okay, as time demands, we have to start with our first session. It gives me immense pleasure to introduce our first speaker. Before joining, actually she has done PhD in pharmacy from University College London, United Kingdom in 2012. Before that, she has pursued Master of Science in Drug Delivery, University of London in 2008. She has done a Bachelor of Pharmacy, International Islamic University, Malaysia, Kuantan in 2007. Before joining International Islamic University of Malaysia, she was working as head of department in research and development in IKOP, which is a pilot manufacturing plant. Currently, she is working as an associate professor in Department of Pharmaceutical Technology. She is carrying out various teaching responsibilities. Till today, she has taught approximately 10 to 12 subjects, which contributes community pharmacy practice, cosmetics and cosmeceuticals, then disperse system, dosage designing 1 and 2. These are some examples of its subject. She has developed a new course, cosmetics and cosmeceuticals in 2016. Approximately 40 research articles were published by her till date in renowned publications. Also, she has written 3 books. She has received 10 to 12 research grants. She has received 42 national and international presentation. She has presented herself. In 2019, she was awarded as Rising Star Scholarship Award from the World Congress Dermatology, which was the international award. She again, before that, she was awarded with the National Award in 2015 for the Best Poster Award from Malaysia Pharmaceutical Society. A research award contributes to in 2015. The topic was silver microsponges, a novel carrier for resecvimode in treating skin cancer in the International Research, Invention and Innovation Exhibition, which was held in Malaysia. Then silver cocoa butter, efficient moisturizer and anti-aging in International Research, Invention and Innovation Exhibition, which was held again in Malaysia. So this was the award and the very well-known personality in the field of pharmacy. I welcome Dr. Hazrina Binti Abhadi, Associate Professor from the International Islamic University, Malaysia, and I request her to start with the first session of today's second international conference on traditional medicine.
Right, thank you very much, Mr. Chairperson. Can you hear me?
Ma'am, you are
Yes.
audible. You can
Can you
start.
hear? Okay, I'm speaking now. Can you hear me? I'm checking.
Yes, ma'am, you are clear.
All right. Okay. Good. Right. A very good morning, everyone in Nashik, India. Perhaps you are somewhere else, not in Nashik, but in India. Good morning to you here, 12 noon in Malaysia. So I hope everyone is fully awake. All right. And to listen to my 1 hour talk, I will try to make it interesting since I can see many of the students attending this talk. Right. Before I start, I would like to thank Dr. Yogesh Ushir, the program chair of this ICTM conference, and SMBT Sevabhavi Trust, SMBT Institute of Diploma Pharmacy. Right. Thank you very much for the kind invitation for me to speak today. Right. So let me share my screen. And please bear with me for 1 hour, and then we can have some discussion, hopefully. All right, for this morning topic, as the title I put in the abstract is about the ancestral wound healing treatment in modern application. Right, for this ancestral wound healing treatment, actually the compound or the treatment that I'm referring to is the one that we always consume in our diet, which is honey. Right. So I hope it's going to be interesting talk. I'm going to share with you, first of all, about the history of honey, right? How honey is formed, just briefly, and different types of honey. And then we see how the roles of honey in wound healing. And then last but not least, but I will share with you guys recent findings from my research team before I conclude the talk for today. Okay, so honey actually it's used currently and also actually was used since a very long time ago. And if you can see on my slide here, the photo there is actually a rock painting. What is actually called as the Man of Bicop, which actually was found in Spider Cave in Valencia, Spain. Even though it was found in 1921, but the archaeologists believed that this painting actually existed since Paleolithic time. So it shows here that the human near to the beehive, and you can see all the bees surrounded him. So what actually this painting tried to show is, even though the man has to face the challenges to gather the honey, but they still try to get the honey as part of their diet. However, archaeologists couldn't find that time any evidence to show the use of honey for medicinal purposes. All right. Sorry, if the host can remove me from being the co-host, because like people keep popping out. I mean, I get the notification to allow people to enter the session. Okay, thank you. Right. So archaeologists after that found this painting, right? You can see on the wall. Okay, what it's trying to show here is actually to see they interpret this as a life cycle of the bee in honeycomb. So the uses of honey during this time, this is Neolithic era, because they found this in one shrine, Neolithic shrine at Kettle Hill, and they believe that honey during that era were used as a natural sweetener to treat the wounds even back then, and they used the wax for painting, embalming bodies, as binder for ships and boats, and as well as for cosmetics. Right. And a few thousand years back, honey also was mentioned in mythology, in myth, Greek myth especially, and Egyptian myth. And if you want to go and read about the Greek mythology, they mentioned that about Zeus, that was hidden from his evil father and was raised only on milk and honey to make him strong. And then later on, he managed to take the throne from his evil father. And as for the Egyptian mythology, honey was used actually as offering to the gods. to show their devotion and worship. And in fact, archaeologists found jars of honey in the tombs of Pharaoh because they believe that honey can be sustenance for the next life. All right. So for formation of honey, we know like one big beehive, and we wonder what's going on there, how actually honey is formed. So basically, each beehive, they only have one queen bee, a special queen bee, which is actually the only female in that beehive that is sexually developed in the hive. And the main role of the queen is only to lay eggs after mating with the drones, right? So in one beehive, we have only one queen bee, and we have about 500 to 1,000 drones. And these drones is actually the male bees. And their purpose, the function of these drones is actually just to mate with the queen. And the worker bees, which is about 30,000 to 60,000 worker bees in one beehive, and they do all the job. They are actually the underdeveloped females, the sexually underdeveloped females bee. So what they do everything else, okay, like collecting the nectar, caring for larvae, cleaning the nursery, and making wax for comb. So basically they have to go out and collect the nectar from the flowers, and later in the hive, they have to ingest. So basically they ingest the nectar, and then the nectar will be later digested and reach certain level of quality. So the nectar will be stored in the honeycomb, but because it contains a lot of water, high amount of water, so again, the drying process will take place and will be done again by the worker bees. And how they do it, they will fan their wings to assist evaporation of water. Only then the honeycomb will be sealed with wax in order to preserve the honey. So that's when we get the wax in the honey. All right, how about the composition of honey? Of course, it's sweet, so it must have some sugars in it, right? The consistency of honey varies. Okay, first of all, it varies depending on the source of nectar, and this source of nectar actually varies depending on the flowers as well. So the main point here I want to highlight is actually, regardless of the sources of the nectar or the different types of flowers, but the major constituents of a honey remain constant. So meaning that every type or any type of honey, of course, they would have sugars, they will have proteins, vitamins, minerals, and this peroxide component, and as well as non-peroxide components. All right, as for sugars, all right, the carbohydrate this is the carbohydrate actually present the main carbohydrate, glucose and fructose. Okay, and but on top of glucose and fructose, they also have isomaltose, turanose, and maltulose. Okay, so basically it's a complex mixture of sugar, and this complex sugar, even though they are complex mixture of sugar, but they can be immediately digestible. Right, and they have various vitamins in honey, like vitamin B6, thiamine, vitamin C, folic acid, even they have. And as for the minerals, they have iron, magnesium, calcium, and also a few different amino acids such as proline and phenylalanine, which is very important as antioxidants. Right. As for the gluconic acid, actually is the product of the enzyme glucose oxidase and glucose itself. And the enzyme in honey, especially if later on we go to the stingless bee honey, is a little bit acidic. And actually all honey has also enzymes, right, which actually can be divided into two types, which are one originated from the worker bees. Remember just now I mentioned that the worker bee has to process the nectar that they collected. So the enzyme originating from hydropharyngeal gland of the worker honeybees, like invertase, glucose oxidase, amylase, catalase, and acid phosphatase. And for the enzyme originating from plants, okay, like catalase and acid phosphatase, and as well as small amount of amylase. And all these enzyme, especially glucose oxidase enzyme, is actually very important because it produces hydrogen peroxide, which not only help to preserve the honey, right, but also it has antibacterial activity. Right. As for the different types of honey, depending on the flower, and there are a lot more. If in Malaysia itself, we also have tualang honey, which is actually wild, the honey that we can get from the jungle. Many people do research on this tualang honey. But the one that I would like to share to you just on the acacia honey, manuka honey, and stainless bee honey. The reason why I'm sharing this, acacia honey, very well known, used in cosmetics field for cosmetics products because it has a very high activity as moisturizer. It's actually honey can act as what we call it a moisturizer because of its natural humectant properties. So it's contributed from the high content of glucose and fructose in honey. And how this glucose and fructose actually helps in the mechanism of giving hydration to the skin is actually this fructose and glucose can form hydrogen bridges with water to retain the moisture in the horny layer skin layers, and therefore can provide some hydration to the skin. And if you see manuka honey, right, is widely used and studied in for dermatology application. And in fact, if you go to the patent search, there are so many products for dermatology, and they use manuka honey. So you can go and read. This is one of the example of the studies done using manuka honey, but it's for me it's very established because for us here in Malaysia, we want to do research, we have to do something new. So that's when it comes to my interest, which is stingless bee honey. Right. I'm not sure in India if you have stingless bee honey, but for us here in Malaysia, is actually we can grow even stingless. We can have our own stingless bee farm because, as its name, is stingless. It doesn't sting. So this stingless bee honey, right? We can have standardized source. Basically, we can ensure that there will be less variation. If we use, say, wild honey like tualang honey, another local honey in Malaysia, then there will be some variations in terms of the composition of the honey. In terms of percentage, of course, the composition, namely like glucose, the enzyme, vitamins will be still there, the phenolic compounds and whatnot. But then in terms of the percentage of the composition varies depending on the source of the nectar. Right. So my research group, we do few studies, right? Few research using stingless bee honey, and one of it is for wound healing. Right. The reason why you can see here, right, or you can read our full paper. I put a name there. Is Jalil Kasmuri and myself, Hadi. Stingless bee honey, the natural wound healer, review paper. Before we started the project of using stingless bee honey into hydrogel for wound healing. So from the literature, it has been found and established that stingless bee honey has broad spectrum of antibacterial activity, and it has immunomodulatory characteristics. It's not just anti-inflammation, but it's immunomodulator, which is very good during the wound healing, which I will explain later. And also it contains a lot of high amount of phenolic compound, flavonoids and enzymes like glucose oxidase and catalase, which all these compounds associated with the antioxidant activity. So basically, this immunomodulatory, antibacterial, antioxidant activity, and as well as in general honey as humectant, which can provide or act as moisturizer, all these criteria actually the roles of honey in wound healing and and also very important to have during the wound healing process. So let's see the function or the roles of honey in wound healing. As I mentioned just now, honey is antioxidant, anti-inflammatory or immunomodulator, antimicrobial, and can be used as moisturizer. Right. Honey as antioxidant. Okay, there are 4 stages altogether in the wound healing process, but I would like to bring your attention during the beginning of the wound healing. So when the wound just be induced to the skin, right? So when the hematosis and inflammation take place. So basically in the hemostasis stage, which is the beginning of the wound healing process, vasodilation take place, okay, around the wounded tissue, and overstimulation of vasodilation, right? Because the inducible nitric oxide synthase, which actually will lead to the production of hydrogen peroxide and other reactive oxygen species, ROS. Okay, so we do not want reactive oxygen species, ROS, because this accumulation of ROS will not only harm the wounded site, but if we leave it, it actually will spread to other organs in the body. So we want the antioxidant, and as I mentioned just now, honey has antioxidant activity, and there are studies and shows in Malaysia, the one team, the researchers from Margi is Malaysian Agricultural Research and Development Institute, a very big research institute in Malaysia. They identify these 2 compounds, PCA and 4-hydroxyphenylacetic acid. PCA, protocatechoic acid. Okay, and this PCA actually we can even buy from the market. It is very well known, very strong antioxidant, and stingless bee honey has very high concentration or high amount of PCA in stingless bee honey compared to other honey. So what a PCA does, because as a strong antioxidant, so PCA improves the cell proliferation in the wound healing process, and this 4-hydroxyphenylacetic acid can help to scavenge those reactive oxygen and nitrogen species. So it can be used as a good antioxidant, stingless bee honey. All right. And honey also can be used as antimicrobial, and the property as antimicrobial also critical or important in wound. Why? Because in any wounds, microorganisms already there, but they usually do not infect the wound, usually, and the wound also will heal. But this situation, okay, when there is no disturbance, okay, in terms of the equilibrium of the immune systems and the bioburden of the wound area. However, sometimes bioburden overcomes the balance, so they will impair the wound healing process. So once the impairment of the healing process takes place, the bacteria will proliferate and occupy the host tissue. So this invasion, basically, of foreign bodies into wound will hinder or delay the healing process and can cause formation of granuloma or an abscess. So if you just leave it, okay, after a while, there will be another problem comes, okay, not just the wound, but like the leftover collagen produced, because it's going to be a very prolonged or long period of wound healing, then the patient will have keloid scars, okay, and it's difficult to remove keloid scars. There are a lot of other studies in the literature shows that stingless bee honey has the ability to inhibit both growth positive and gram negative bacteria. Okay, how this antibacterial or antimicrobial presence shows in honey is actually comes from 2 components of honey. Antioxidant just now, it's coming from the phenolic compound of honey. And as for the antimicrobial, it's coming from both peroxide and non-peroxide components of honey. So for the peroxide component is based on the activity of the hydrogen peroxide, H2O2, which actually regulated by two important enzymes, glucose oxidase and catalase. Right, so glucose oxidase actually induce the production of hydrogen peroxide, and the catalase destroy the hydrogen peroxide. So some balance in order to preserve the nutritional content of honey, and this hydrogen peroxide also will enhance the production of cytokines for the inflammatory response to kill the bacteria. As for the non-peroxide, are based on the phytochemicals, okay, high sugar content and the acidity of the stingless bee honey. If you can find stingless bee honey in India, you can compare the taste and even the smell. It has a bit strong smell, acidic smell to stingless bee honey. Right, so these phytochemical component in honey actually contribute to the effects, antibacterial effects like flavonoids, phenolic substance, of course, and also antibacterial peptides that present in honey. And these components it's by inhibit the phagocytosis, so it's prevent the superoxide free radicals from damaging the tissues. And about the acidity, because most of the bacteria, microbes, they like to grow in the neutral pH between 7.2 and 7.4. So this kind of microbes will be suppressed by stingless bee honey because they are more acidic in nature. All right, the next role of honey is anti-inflammatory or immunomodulator, right? Is actually contributed by the phenolic compounds. Okay, inflammation, okay, is a good response from our body when we get hurt, because it's actually is a protective effort by our body to remove any harmful agent and to start the healing process. Okay, and you can see here macrophages. Okay, I hope you can see my cursor. Macrophages here would start to encourage or increase the production of pro-inflammatory cytokines, the nitric oxide, and PGE2. And all these inflammation mediators actually highly cytotoxic and are involved in innate response by killing the target cells. So even though these mediators, okay, of course it has beneficial effects. That's why it's activated by macrophages to protect the body, but it actually can cause tissue damage, and this can lead to chronic diseases and also will impair the wound healing. So the introduction of anti-inflammatory, okay, or the properties of anti-inflammatory in honey can actually help to control overproduction of these cytokines or the inflammatory response. So it's not going to damage the skin cells and not going to delay the wound healing process. Right. So in earlier period, the emphasis of wound care management, okay, to have the wound area dry, to preserve the dryness around the wound. However, since 2003, okay, clinicians together with researchers, they agreed on new standard wound care strategy, whereby they want to have the wound area to be moist, not wet, but moist, which following the acronym of the concept of what they call it as TIME, tissue, infection or inflammation, moisture, and age, right? So why they want the moist area? Okay, it's not dry, it's not wet, but we have to keep the wound area moist. Basically, a moist surrounding is required to prevent secondary infection, one, and two, actually can create effective oxygen circulation. So because moist condition also can impede the desiccation and necrosis, can help to enhance the formation of the blood vessels and connective tissues, as well as it can help to rehydrate the dried out tissues. So application of moisturizer to the wound actually okay, can also helps to reduce the possibilities of the appearance of scars. On the other hand, because we need to avoid the wound dryness, because dry environment actually can delay the migration of the epidermal cells, because as you know, the what we call it, the epidermal cells, especially the daughter cells, daughter skin cells from the stratum basal, the lowest layer of the epidermis layer, is going to take like 14 days, isn't it, to reach to the outermost layer of the skin. So it will delay the movement of the new cells. So we do not want to delay that. We want the new cells to form so that can repair the skin. So we want to provide the moist environment, or the ideal environment for the skin cells to be generated, right? And also the can reduce the scarring. Okay, so the high water content in stainless behind also can prevent the dehydration to the wound because of the osmotic effect that gradually delivers fluid to the wound tissue. So we can maintain the moist environment around the tissue, okay, accelerate the healing, and we can give a continuous moist condition, okay, but we have to be careful if keep giving the most, what we call it, hydration, the wounded area can be wet. However, with the application of honey, because honey has high osmolarity, so it's not going to wet the wounded area, but only can just provide or maintain the moist surrounding. Right. So since like I mentioned before that, even in Neolithic area, okay, honey has been used for the treatment of wound. The idea of the project actually, because when we see the clinical practice here in Malaysia, even the orthopedic surgeons or in the internal medicines, they use honey sometimes to treat the wound, but they usually sometimes they just use, or even the people, they just use honey and they put directly on the skin. So the application of honey on wound tissue like that, okay, is not going to be reliable, because the tissue will absorb the fluid, obviously, and also the dose of honey applied obviously not consistent. And as a pharmacist, we always looking into the dose consistency as well. And besides that, when we put honey, just imagine honey in our kitchen, we put on the skin, so we need something else to cover. So for those who are suffering really, like in the chronic wound patients, for example, in the diabetic foot patient, or like if you have large area of the wound, then you put honey, and obviously you will put some bandage or gauze to cover it, and then you have to open and do the dressing, and then put the honey again. And this application actually can cause non-compliance in patient, okay, not just we have the problems, dose inconsistency, but also patient non-compliance, because the patient may suffer from the pain during the dressing changes. So that's when we are thinking to use this ancestral wound healing treatment. We want to incorporate into a modern application or modern dosage form, which is hydrogel. Why hydrogel among all other dosage forms, like the gel, the normal gels, or the cream? Because hydrogel can, what we call it, can provide, give the texture of similar to the granulation tissue, and it's very cooling, provide a soothing effect, and can also helps to reduce the pain. All right, so let's share with everyone here the findings from my research teams. The project or the data that I presented here actually done by this gentleman here, Dr. Azri. He just recently graduated with a PhD from the group. Right, so when we started the formulation of honey hydrogel, because there are so many excipients that we can use to have hydrogel, and also there are a lot of optimization need to be done to ensure that we get a stable, right, and good release of honey from hydrogel. So we get some helps from design of expert is software, right? So now in my research teams, most of the formulation we use design of expert to help us to simulate and predict what are the factors that may actually influence the formulation. So we use for this particular hydrogel, okay, we use screening factors, two-level factorial designs, and then after that we use optimization of the formulation using the central composite design. Right. So the RSM by design of expert, okay. So we identify or we put the 5 factors first, all right. And we decided from the literature to have PEG, agar, and glycerol, okay, as the factors influence the hydrogel, and as well as the temperature and cycles, because we use freeze and thawing method to produce this hydrogel. So how we prepare the hydrogel? Solution A actually containing water and polyvinyl alcohol. B containing the PEG, agar, and PEG, agar. Another one is the I cannot recall, glycerol. Okay, and then after that we mix these two solution and we submerge in ultrasonic water bath to remove the bubble, degas it, and then we cool down the mixture to about 40 degrees, and then before we add honey. And then after about 45 minutes, we distributed those hydrogels in the Petri dish before we subject the formulation to the freeze thawing method. So basically we freeze the mixture for 18 hours at -20. And then after that, we take it out and put it at 25 degrees Celsius to thaw. So it's complete one cycle, 24 hours, and these cycles were repeated 3 times. All right, so for the response test, because we're using the design of experiment, DOE, so among all characterization, we use the swelling ratio as the response test to study the influence of the selected factors, just now the 5 factors, towards the formulation. Right, and from the analysis, we need to get the p-value less than 0.05 in order for us to conclude that that particular factor affecting the formulation. So as for my formulation, this hydrogel formulation, we found that the concentration or the amount of PEG, agar, and the cycles would contribute to the or affecting the formulation, the hydrogel formulation. Right, and Pareto chart also given by the DOE, right? So this actually to help us to understand further how those factors we said is affecting the formulation, but how it affects the formulation, whether it's going to give the positive effect or negative effect, it depends on the what we call it, the bar here. The orange bar here actually give the denotes, meaning it tells us is give the positive influence, and the blue bar give us the negative influence. So meaning that A is the PEG, polyethylene glycol, and E is the number of cycles. So basically PEG and number of cycles give positive influence to the swelling ratio of the hydrogel, and meanwhile agar give negative influence to the swelling ratio of the gel. Okay, so then we do the optimization of the formulation. Right, there are 3 factors just now, okay, the PEG, agar, and the cycles. However, since the cycle during the because we also have to do the concurrent preparation in the lab as well, so we fix the cycles to 3 cycles only, because of the we cannot go beyond that, because it will be too time consuming. So we only fix, so basically when we do the optimization, we fix the cycle and we try to do the optimization using the software by modulating the two factors only, PEG and EGAL. Right, so the system helped us and provided us with these 13 formulations. And using the swelling ratio as our reference, okay, for the characterization for the hydrogel. And if you remember Pareto chart just now, EGA give the negative influence to the swelling ratio. So basically we can see that with the increased amount of EGA in the formulation, it's actually reduced the swelling ratio. And on the other hand, since PEG influence according to the Pareto chart previously, so polyethylene glycol have positive impact on the swelling ratio. So we can see here that what we call it, the PEG, the increased amount of PEG in the formulation would increase the swelling ratio of the hydrogel prepared. So these are optimized formulation, then after that generated by the DOE. And so we concluded that we go for the full characterization of hydrogel using these formulations. Formulation 1 actually is the control without honey, and formulation 2, 3, 4 is with honey. Right. There are few other two, I think, yeah, antimicrobial testing and a few more in vitro studies that I'm not putting here because I don't think I will have time to share everything. But these are the main characterizations that we did for the hydrogel that we formulated in the lab. So which is swelling ratio, WVT test or TR, gel fraction, rheological properties using rheometer, FDIR, release studies, and stability tests. All right, for swelling ratio from the figure, we can see that formulation 2, 3, 4, which is the, these are the formulations of hydrogel honey, has higher swelling capability than control formulation. So what does it tell us? It's actually shows the, it's measured the capacity of the hydrogels to absorb amount water, right? And then we also do the gel fraction. And generally for gel fraction is indicate high percentage of gel fraction is indicate strength of the gel. But then we, because as we know, high strength of gel will have reduced flexibility. So we would like to have some balance between the what we call it, the strength, gel strength, and also its flexibility, especially when you want to apply this on the, if you want to apply this on the chronic wound, right? Because we do not want it to be ruptured or to being too rigid, the hydrogel. All right. As for the water vapor transmission rate, okay, in WVTR, we can see from the figure, okay, is no significant difference. Okay, the ideal hydrogel dressing actually to have good ability to control water loss and to maintain moisture. So we high WVTR actually can cause wound to dry more rapidly than normal and can lead to scar formation. So we want to have intermediate WVTR. Okay, so and comparing the formulation without honey, so WVTR of for the formulations containing honey is lower, even though it's not a significantly difference, but it's lower than hydrogel blank without honey. Right. As for the rheological properties, we measure the LVR, which is the linear viscoelastic region, and through the oscillation stress strip test. So we want to see the what we call it, the shear rates for the stress strip test. Right. So we see here, okay, for the LVR, the based on these rheological properties, okay, you can see formulation 3, okay, has the best rheological properties among others. Why I said it has the best properties among others, because it doesn't have the difference between the formation 2, 3, and 4 is actually 3 doesn't have glycerol. And why I said it's good rheological properties compared to other formulations, because it has better and clearer this LVR line. All right, FDIR, okay, to see whether there is interaction between the honey and the excipients used in hydrogel. So for the, what we call it, the excipients compatibility or interaction, we can use, of course, FDIR, also DSC. We can see the thermal changes if we want to, but we didn't do it here. We do FDIR, okay, to see the fingerprint of the excipients or the formulations we have here. Right, the broad spectrum, if you can see here, the broad spectrum at the 3,200 to 3550 here is actually related to the hydroxy stretching. Where's my cursor? Okay, hydroxyl is related to hydroxyl stretching, which indicate actually the presence of water. And this hydroxyl actually is the functional group of the polyvinyl alcohol, PVA, that we use, okay, in the mixture A just now. And actually, hydroxy group also present in PEG. So that's why we have this broad band from 3,200 to 3550. And then if you compare A, B, C, D, A is actually formulation without the honey. Okay, this hydroxyl group band, it doesn't change even with the, when we add the honey. So basically, there is no substantial interaction observed, okay, in comparison between the honey gel, honey hydrogel with the blind hydrogel. All right, so we have now honey in the hydrogel, but we also have to make sure that honey can leave the hydrogel and go to the site, the targeted site. So we did the in vitro drug release using total immersion method, and then for 24 hours, and we analyze using the UV spec. So what we observe is that the release of these of honey actually higher in the beginning and then slowly, okay, so because this is the cumulative release, right, the y-axis. So this actually, from the practical point of view for wound dressing, this sustained release is desirable, because in the early stage of the medicated hydrogel, the release should be rapid, right, the release should be rapid, and then followed by period of sustained release of the drug, so that the application of the drug on the wound will be sufficient. And then after 24 hours, just at 24 hours, we have a complete release of honey. So this is actually very useful and very practical, because we do not have to do the changes, because we can maintain the honey on the wound skin, the wounded skin, for 24 hours without changing the hydrogel as well. Excuse me. Right. So based on this characterization, and then also we do the stability studies, then we proceed further later to choose which types, which formulation is the best to try on the animal model. But before that, we subjected all the formulations to the stability studies. But since this hydrogel has to be kept in the refrigerator, basically, so about 4-5 degrees Celsius. So according to the Asian guideline on stability studies of drug, so we considered by storing the samples at 25 degrees Celsius with about 50 relative humidity for 3 months is considered as accelerated. But if the normal stability studies, this is the we call it real-time stability studies. But because hydrogel will be stored at the, in the refrigerator at 5 degrees, so this is considered as accelerated. So after 3 months of accelerated stability studies, we again characterize the swelling ratio and gel fraction properties if there are any significant changes. And fortunately, okay, there is no significant changes, slightly decrement, but still fall under the what we call it, the guideline. Still can be reported as stable. Right. So from the in vitro study just now, the relay study, from the characterization, then we choose honey hydrogel formulation 3 as our product, right? And for the in vivo efficacy test, we use rabbits, and we have 3 groups, no treatment, and then 1 group we apply only hydrogel, exactly the same formulation but just without honey, and then another one honey hydrogel. Right, we use rabbit instead of rats because we want to reduce the number of animals sacrificed at the end of the study. So we use rabbits, and we do multiple incision actually on the rabbit's back to cover the 3 groups. Before this is actually our product just now. Let me go back. Here on the photo on the right here, the top right, this is the hydrogel that we have at the end of the formulation, and this is the one that we put on the in vivo studies on our rabbits, right? So what we did actually during the animal testing, we shave the dorsal thoracic region of the rabbit, and then we gave IM injection of ketamine to anesthetize the rabbit, ketamine with xylazine, and then we cleaned and disinfected the area using 70% ethanol, so to make sure there is no introduction of the any bacteria or to prevent infection. Because then right after that we use biopsy punch and we cut the rabbit. So we make like about 4 mm diameter incision wound on the rabbit. So the wounding day itself, we consider it as day 0, and the wounds were categorized as no treatment, blank hydrogel, treated with blank hydrogel, and treated with honey hydrogel. And we treated the wounds OD, since the release study shows at 24 hours, the honey release from the hydrogel was 100%. And at the predetermined level intervals, we capture the wound image using the video scope probe of DermaLab combo. And then we measure the size of the wound closure. Right? Okay. At the end of the study, so we continue the studies and we keep observing, and then we notice day 9, okay, 1 group, the treated group is completely healed. So what we did after 9 days of treatment, we sacrificed the rabbits by carbon dioxide overdose, and then the wound tissues were extracted and we fixed them immediately with the formalin, and we subjected them to histopathological studies to observe the changes at the microscopic level to support the in vitro data. And we use the HNE, hematocillin and eosin, HNE stain, and as well as Mallory trichrome. And we also use normal skin tissues, and we stain them, also the normal skin tissues, with HNE and Mallory as baseline for comparison with the pathological changes on the wounded tissue. So we use what we call it different types of the HNE is the standard staining, and Mallory's trichrome stain because we want to see to get clear visualization, because we cannot just simply repeat the experiment, so we just do these 2 stainings, because theoretically Mallory's trichrome stain should give us a clearer visualization of the collagen fibers, because collagen fibers production should be increased during the wound healing process. So both stannings we perform using the guideline. As for the HNE, we follow the Sejipath Leica Microsystem USA, and for the Mallory's trichrome staining, we follow the protocol outlined by the Diapath Spa from Italy. Right. So we can see here the honey hydrogel groups, okay, display significant increase in wound closure percentage, right? Even started from day 3, they are significantly different compared to the blank at the dark blue here, and this one is the untreated, right? And further, even to the day 6 to day 9, still they have the significant difference in terms of the percentage of wound closure when we do the calculation in terms of the wound closure, and these supported by the histological assessment, right? I cannot compile all the 4 photos together. It's actually we have the normal tissue, normal no treatment, right? For the histological assessment, blank hydrogel as well as honey hydrogel. So, but I have to divide because otherwise I cannot really show the photos to you. So here as for the normal tissue, we can see the epidermis is relatively thinner, right, compared to no treatment group. Okay, the epidermis layer is thick. And another observation you can see is the presence of many skin appendages, right, can be observed in the normal tissue but not in the no treatment tissue, right. As for the blank hydrogel, again, you can see that the skin epidermis still thick, right, compared to the one treated with honey hydrogel. Okay, so to convince you further, I put side by side the normal tissue, okay, staining, HNE stain normal tissue compared to the honey hydrogel. So we can see here that the thickness of the epidermis is more or less the same. And to further support our finding, right, we also have proof in terms of the visual gross observation, where we use video scope image, which actually generated by the Malek combo cortex technology, a very useful method. We have different probes of the Malek combo in our lab. So one of it, we can use the video scope probe to observe and capture this image to really see what's happening on the skin. So as you can see here, at day 9 for the hydrogel with honey, the wound completely closed and healed, and compared to the hydrogel alone, and of course no treatment, the rabbit suffered a little bit, right. So in conclusions, I can say that stainless bee honey-based hydrogel demonstrated promising healing properties, and the results suggested that stainless bee honey-based hydrogel can promote full thickness wound healing by promoting the wound contraction as well as collagen formation, and implies that introduction of stainless bee honey hydrogel provides synergistic effect to enhance the rate of wound healing, because when we compare the blank hydrogel versus the honey hydrogel. Okay. Right. So the output from this research project is actually we already filed the patent, still ongoing before it's granted. So the data is now already filed, and we also came out with 2 trademarks for the products. One product, Madhu, is for the cosmetics application, and we have Hydrobi products is for the dermatology or pharmaceutical application. And of course, I need to acknowledge the funder for the project is Ministry of Education Malaysia, and endless support from the university to help me to file the patent, get the trademark, and so on. And of course, I wouldn't have the data, even just we have the funding, but I wouldn't have the data without the support of the work done by my team. This is my group, and we are at the Faculty of Pharmacy. Sometimes, actually, the photo here is from the progress meeting, and we had fun, of course, and this is birthday celebration. We work hard, we play hard. And where we are in Kuantan, Pahang, in a bit, not really far from KL. It's about 230 kilometers away from KL, or we can enjoy 2.5 hours driving from KL, or short flights, about 40 minutes flight only. You cannot even sleep during the flight, and we reach Kuantan from KL. All right, and here are some of the photos of my place. I would say very peaceful, beautiful city. This is not my hometown. I'm actually from East Coast of Malaysia. I'm from Kelantan, not from here, but I studied here 2003, 2007. So it's still the same, except they have more resorts here. And the new Starbucks are one of the prettiest Starbucks in Malaysia. All right, I think that's all. Thank you very much.
Thank you so much ma'am, for a wonderful session. Welcome to the delegates. If you want to ask any query, any question regarding this session, then you are open for this session.
Or you can email me as well if you want to. My email is hazrina@iium.edu.my. If you need papers or any explanation, I would be more than happy to help.
Yes, sure, ma'am. Anyone have any doubt regarding this session? The session was really fruitful, and ma'am has explained us really well in an easy manner what she has done. At the end of session, I would like to ask program chair of ICTM 2021, Dr. Yogesh, to share kindly give a vote of thanks.
Thank you.
Namaste, Professor Hazrina, namaste. So on behalf of second ICTM organizing committee, on behalf of SMBT Seva Bhavi Trust, I really thankful to the Professor Hazrina ma'am who accept the invitation with my single mail, and we are being in touch from the last 2 months, and the things are happen very nicely over here. We say that your session is very fruitful for my delegates as well, not only for Indian delegates, but the international delegates also enjoy your session. I just observing all the things from the computer also. So your session is really beneficial for them, and I'm very thankful once again, ma'am. Be in touch with us. Thank you very much. Namaste.
Thank you so much, sir, and thank you very much, ma'am. We look forward for your physical presence on the next ICTM 2022. Thank you so much. Now here I would like to conclude this session, and we'll have a next session at sharp at 11 o'clock. Thank you so much. I hope everyone is joining. Now the next session for the second ICTM, we are having speaker from the Thailand, Dr. Frederick Eze is an academic special expert at the Faculty of Pharmaceutical Sciences, Prince of Songkla University, Hatyai, Thailand. He obtained his BSc degree in biochemistry from Joseph Ayo Babalola University, where he graduated at the top of his class. With an abiding interest.
Sorry, this one. Hold on. Yes, speaker, yes. Louder.
Hello, Professor. Am I audible to you? Professor Eze, am I audible to you?
You're talking to me. I can hear you clearly.
Yes, I'm talking to you only, and I'm introducing you to all the delegates.
Okay, it's a pleasure. I really appreciate it.
Yes, thank you so much. With an abiding interest in the understanding the therapeutic potentials and molecular underpinning of bioactive from the plants, he obtained MSc and PhD degrees in biochemistry from the University of Benin City, and Prince of Songkla University, Hatyai, respectively, whilst focusing on the unraveling the therapeutic basis of local medicinal plants in combating chronic diseases. Dr. Eze's investigation also pertains to chemical biology, development of novel enrichment methods for the active ingredients, biomaterials fabrication, as well as drug delivery systems. His work has been published in highly reputed scientific journals, including Food Chemistry Journal, Material Science and Engineering, and Biomolecular journals. So I welcome Frederick Eze, and I request him kindly start with his session. As all delegates are awaiting, please go ahead.
Thank you so much. And it's a pleasure to be here. And I count this a remarkable privilege to be joining you in this wonderful conference. First, I would like to extend my utmost gratitude to Dr. Yogesh for inviting me to be part of this tremendous event. And I'm indeed truly grateful. Please, can you upload the slide, the PowerPoint presentation? Okay, so while we are in a very troubling time, if you look at the news everywhere, you see concerns about the current pandemic, which is the reason why we are not in India today. Some of us, although we would have loved to be there, but because of the current global pandemic, we couldn't join you physically. And this pandemic has claimed the lives of over 1.9 million people as of today, which is a terrible thing to humanity. It's indeed a great tragedy. But I have, please, the next slide. I have some good news for us, some not so good news for us, and also I would like to also have more good news for us. So I'll start with the good news. And here's the good news. Regardless, or I would say in spite of the current global pandemic, which is COVID-19, and the tragedy that it is causing all around the world, if you look at the data, at least based on what we have been provided from the World Health Organization in the global health statistics of the recent year, 2019, we can see that diseases like COVID-19 are not having the edge. They are not winning. As a matter of fact, if we look at the list of the 10 top causes of mortality, the 10 top causes of mortality in the year 2019, we can see that on that list, non-infectious diseases, or rather infectious diseases like COVID-19, are not the most prominent on the list of the top 10 diseases that are plaguing humanity. As a matter of fact, if we look at that list, we realize that diseases like TB, tuberculosis, and HIV are no longer on that list, which is good news for us. And the good news is that at least that encourages us that COVID will not have the upper hand in this situation, because the trend, the general trend, shows that sciences and the endeavors of research scientists are making it easier for us to tackle infectious conditions. However, that should not stop us from also taking notice of the other diseases that are plaguing mankind. And in this case, there's something remarkable, which is the fact that non-communicable diseases have been persistent for over 20 years as the top cause of death for over 2 decades. And that is even more remarkable in the fact that just in the last year, that's in 2019, not 2020, for the first time, diabetes was able to make it in the list of top 10 killers. That's what I call that list, top 10 killers of mankind. It's not only diabetes, but we also have Alzheimer's for the first time. Alzheimer's was able to join that list of top 10 killers. So even though infectious and communicable diseases are killing a huge number of people, we must not lose sight of the fact that even recently, as of 2019, the number 1 cause of death on earth are not communicable diseases. Rather, they are non-communicable diseases, such as cardiovascular diseases, such as diabetes, such as Alzheimer's disease. So research efforts have to take cognizance of this. And the question that we ask ourselves is, how do we approach these non-communicable diseases? These diseases that you don't get infected by coming in contact with somebody who already has it. How do we tackle this disease? How do we tackle diseases such as cardiovascular diseases, such as cancer, such as diabetes? How do we approach them? So I would propose that we need a multifaceted approach against these diseases, because there's no magic bullet that we can apply to challenge these conditions. We need a Swiss knife approach whereby we attack these conditions from different perspectives. And given that, the question that arises is there a role for bioactive enriched extracts? Is there any role for traditional medicine in combating these non-communicable diseases? And that is what I'll be focusing on in this talk this morning. Please, the next slide. So if we look at the slide in front of us, we have type 2 diabetes, we have Alzheimer's disease, we have Parkinson's disease, and we also have a rare disease which is called transthyretin amyloidosis. I know a lot of people have not heard of this, but you'll be hearing more of it this morning. So what is common among these 4 diseases? Diabetes, Alzheimer's, Parkinson, and transthyretin amyloidosis. The first thing is that they are all non-communicable diseases. They can't be transmitted like COVID. And they are chronic conditions that develop over a series of time. And as they develop, they become more degenerative. And these diseases have no cure. These are just at the facial level. None of these diseases can be cured. Once a person once these diseases set in a particular individual, then the clock of death starts ticking. Now, at the molecular level, we realize that all these diseases have something in common. In their molecular etiopathogenesis, the way that these diseases come to be, they share a key process in their origin. And that key process is that there is a particular protein that gets misfolded and or aggregates. And once that protein becomes misfolded and aggregates, it transforms from a normal protein into a toxic protein that then starts the pathological process, which then leads to the sickness and the symptoms. And ultimately, in every single one of these, it leads to death. So, please, the next slide. In the case of transthyretin amyloidosis, I would want to use these 4 diseases as to instantiate whether medicinal plants and bioactive enriched extract could have a role in tackling these 4 deadly diseases, because that's what they are. So in the case of transthyretin amyloidosis, just from the name transthyretin is a protein that is produced by the liver. If you look at this pathway, it is released, it is synthesized and released from the liver into the systemic circulation. Under normal physiological circumstances, transthyretin is a very important, it's not very important in its function, but it performs a function regardless. It helps in the distribution of thyroxine. It also helps in the distribution of vitamin A. However, this protein, which in its native state is a tetramer, as you can see, we have 4 colors representing the tetramer, and we also have a small perforation at the center, which is the location where it binds to thyroxine, where thyroxine binds. We call it the T4 binding site. Under normal physiological circumstance, transthyretin is a tetramer. But the tetramer, as we become older and our internal system becomes high in its oxidative burden, transthyretin adapts the tendency, becomes more prone to dissociate from its tetrameric form. It can change into, it can dissociate to release its monomer. While the tetramer is stable, the monomer is not, and the monomer has a likelihood to become misfolded and then forms the toxic aggregate. So once the aggregates are formed, the aggregates have the propensity to attack normal cells in the heart, in the central nervous system, in the liver, in the kidneys. And these proteins accumulate and result in organ dysfunction, which ultimately can lead to death. Both the misfolded oligomers and the mature fibres are capable of inducing cytotoxicity. So please, the next slide. So what are the strategies that have been developed over the years to help to reduce the chances of transthyretin amyloidosis? What are the strategies that have been adopted to slow down the progression of this neurodegenerative disease? So one of the first strategy is to stop the production of transthyretin from the liver. So in this case, we have what we call TTR suppression. That's the name of the strategy. And right now in the market, we have some agents, some drug candidates like inotersen, which help to prevent their small interfering RNAs that help to prevent the production, the synthesis of transthyretin from the liver, because transthyretin is not an essential protein, so to say. So even if we can reduce the synthesis, it's not going to cause any remarkable or substantial physiological changes. So one strategy is to stop their production. A second strategy is to help to stabilize the tetramer. We call that tetramer stabilizers. So if we can stabilize the tetramer and prevent it from dissociating, then the propensity of becoming misfolded and aggregating and producing the toxic fibers and oligomers reduces drastically. A third strategy is to trigger what we call an off-pathway mechanism, whereby even though the monomer is dissociated, we have small molecules that we can use to attach to the native monomer or misfolded oligomers. And by attaching these molecules to these protein precursors, we can remove them from the normal pathologic pathway and divert them into a non-toxic pathway. So then the third strategy here is the fourth strategy here is to attack the fibrils, the mature fibrils. We call this strategy fibril disruption. So if we can help to disrupt the mature fibrils, then we can reduce the cytotoxicity that is caused during transthyretin amyloidosis. So please, next slide. So then, how do we get into this point where we want to see if we can use medicinal plants to tackle transthyretin amyloidosis? I will share you a story, what I used to call my centella asiatica story. You know, several years, precisely 2012, I was still in Nigeria, and I had the opportunity of visiting one of my professors. We used to call him Baba. So I went to his office and we were chatting. All of a sudden, he introduced a product to me. He said, Eze, you have to try this. And I was like, what do you mean, Baba? He said, this is very good for the brain. It can help to improve your memory, can stimulate memory. It can help retention. When you study something, you will not forget it. And that sounds very, very interesting to me. And so I asked him, Baba, what is this? He said, it is centella asiatica. I said, I've never heard of this. He said, yes, I got it from India when I went for a conference. So you see, my centella story actually began in India. And here I'm presenting to you in India. So I got interested. After that discussion, I got interested in centella asiatica and another plant which he also introduced to me, Bacopa monnieri, which he also got from India. This was in Nigeria. So several years later, about 6 years ago, when I came to Thailand, I was engaged in a research project on neurodegeneration, focusing on this particular ailment, transthyretin amyloidosis. I went ahead and proposed 5 plants that can be used to tackle transthyretin, that could be used to tackle transthyretin amyloidosis. Fortunately, 2 of the plants that were approved were centella asiatica and Bacopa monnieri, the plants which Baba introduced to me. So I was so excited, and we got into extraction. We extracted centella. Before I go into that, I would like to even give you a brief overview of centella. It is a plant that is widely common in Asia, just as the name sounds, asiatica. In Thailand, you can find it in almost every local market in the southern part of Thailand. They drink it as freshly prepared juice. They use it in preparing pastries. They put it alongside rice when they are eating it. it is so ubiquitous that if you go to the market, it's so cheap, you can have it anyhow you want it. So it's a common plant that grows in marshy areas and in temperate regions. We didn't have it in Nigeria until Baba introduced it to Nigeria. It's not native, it is native rather in Asia here. So we got into the extraction process where I fractionated the Centella asiatica. I was interested, I wanted to know if Centella will have any effect on transthyretin amyloidosis. So after fractionating it using simple solvent fractionation, I produced about 4 different fractions. I then profile the 4 fractions on the tetramer, the transthyretin tetramer, to see if they could stabilize the transthyretin tetramer. Please, the next slide. So if you look at the figure which you have in front of you, on the SDS gel in your right hand, you'll see that the last lane is the lane that contains the Centella asiatica active fraction. We had 4 fractions. One of the 4 fractions was active. So the last lane contained the active fraction. You could see that the tetramer, the band produced by the tetramer, is thicker, is more intense in its appearance than the closest band, that's the third band. If you look at the third band is a little bit lighter compared to the last band, which shows that you had more tetramer in the last band, which contains our extract, than in the third band, which did not contain our extract. The first band, no, the second band is the control band, which did not contain the stressor, which in this case was urea, which we used to cause tetramer destabilization. So you see that there was no tetramer destabilization here. That's in the second band. But in the third band contained the destabilizer. And you see that a lot of the tetramer were released. And you could also see the corresponding increase in the intensity of the bands produced by the monomers. Then the last lane contains our Centella bioactive fraction. And you could also observe that in this case, the intensity of the tetrameric band was higher. And we then decided to see how Centella bioactive fraction affected the morphology of the protein using transmission electron microscopy. So the image on the right-hand side is the TEM micrograph. And from it, you see that A is the control, which does not contain anything, no stressor, no Centella extract. You see that the proteins are well dispersed and solubilized, and they don't have any aggregates. Then B contains the Centella and the protein. And you can see that in the presence of Centella, the protein still forms some aggregates. Now, in the C part of the figure, you see that even though Centella, the C part does not contain Centella, and you can see that there was a mature fibril that was formed. Unlike in the B, where we only had aggregates and we didn't see any mature fibril. In the C part, we had mature fibril. And the D is just a higher magnification of that mature fibril. So please, next slide. So from this, we quantified, and we also use subsequent techniques like cross-linking, glutaraldehyde chemical cross-linking, ANS probe to determine the binding of the Centella asiatica bioactive. So from there, we realized that Centella asiatica bioactives were able to bind to the tetramer binding site, to the thyroxine binding site. When normally thyroxine binds, we discover that our centellar bioactive fraction, the components there were able to bind at that thyroxine binding site. And by binding to the thyroxine binding site, they were able to increase the tetramer stability of transthyretin by as much as 52%. And because of that, the rate at which the tetramer is able to dissociate subsequently reduced. And that reduction, we surmise, could help to retard the progression of transthyretin amyloidosis. So that was what we were able to take out from this research with centella asiatica, that the bioactive fraction from centella asiatica was able to bind at the thyroxine binding site of the transthyretin tetramer and increase its stability, prevented its dissociation, and inhibited the progression in the amyloidogenic pathway. So that was what we concluded from this. And based on the encouraging results that we had from the centella asiatica, we decided to also test another plant, which Baba suggested to me several years ago. In this case, it was Bacopa monnieri. Please, next slide. So, Bacopa monnieri also is a small plant that is very common in Asia. In India, it's called Brahmi. In Thailand, they call it Promi. And it is widely reputed. Many people know it as one of the herbs that is used for combating neurodegenerative diseases, especially Alzheimer's disease. It's a very, very popular plant in these regards for combating neurodegenerative diseases. Also, some people use it as a nervine to relieve nerve stress. So what we did was we followed the same protocol that we use for centella asiatica. We produced our bioactive fractions, and after producing the bioactive, we fractionated. We extracted, then fractionated, then screened for the bioactive fraction from Bacopa monnieri. But in this case, we decided to see the effect of Bacopa monnieri on two types of transthyretin, because we have the normal wild type human transthyretin, and also we have the mutated form of human transthyretin. And the reason we did this was because the mutated form of human transthyretin is more aggressive in its pathogenicity. So we wanted to see if components from the extract of Bacopa monnieri could help to reduce or even prevent the progression of transthyretin dissociation. Please, next slide. So using our stability experiments, we were able to establish that the bioactive fraction for Bacopa monnieri was able to increase the tetramer stability of both the wild type and the mutated type transthyretin. And in the case of the wild type human transthyretin, Bacopa monnieri enhanced the tetramer stability by 44.2%. But it didn't only enhance the tetramer stability. We discover also that in the presence of Bacopa monnieri, the formation of mature fibrils and oligomers were also impeded, which was quite remarkable, because in the case of centella asiatica, we discovered that the bioactive fraction from centella was only able to stabilize the protein, but it didn't prevent the formation of the amyloid fibrils. Once the protein dissociates, it ended up forming fibrils. But in the case of Bacopa monnieri extract, we realized that even when the protein is able to dissociate in the presence of bacopa, the subsequent stages were also impeded. Please, the next slide. And that result was quite encouraging for us. And based on what we saw with Bacopa moneri and Centella asiatica, and the higher amount of tetramer stabilization that we observed when we used Centella asiatica bioactives, we decided to see if we can identify the active ingredients, the different metabolites that were present, bioactive metabolites that were present in these extracts. So that the idea was to see if we can even raise up the effectiveness beyond what we observed. So in the case of Centella, we used LCMS to profile the compounds that were present, and compounds such as chlorogenic acid, acetoicoside, madecassoside have not been investigated. There were no reports on their anti-amyloidogenic effects when it comes to transthyretin. Also, Centella contained other compounds such as catechin and quercetin and apigenin, but those compounds had already been reported to have some effects, some therapeutic effects on transthyretin. So we decided to look at the compounds that had not been reported, but which were also majorly present in Centella. So what we observed was that these compounds didn't have any substantial effect on transthyretin amyloidosis. That's the newly identified compounds such as chlorogenic acid, acetoicoside, madecassoside. They did not have as much, they didn't even have any significant effect compared to the extract. And that to us was a little bit interesting and unexpected until I stumbled on an article that was also published almost the same time by Chaudhry et al., where in their own case, they use extracts from Juglans manchurica, which is a plant common in Japan. And the methanolic extracts were fractionated to give butanol extract and ethyl acetate extract. These extracts, they discovered that the extracts from the fractions from Juglans were able to inhibit, not inhibit, they were able to dissociate the mature fibrils and thus reduce the cytotoxicity. And because of that positive result, they also wanted to identify the compounds that were present in Juglans manchurica. After realizing that most of the compounds were naphthaquinones, they elucidated the naphthaquinones that were present in Juglans manchurica, and then tried to see if those compounds, those naphthaquinones were able could inhibit, could disrupt the mature fibrils. And they had the same observation. They realized that the naphthaquinones that they identified in their bioactive fractions did not have any effect on transthyretin. Then they now decided to look at other species of Juglans and other compounds. And finally, they were able to identify one particular naphthaquinone in a different species of Juglan. And that naphthaquinone was called Juglone. Juglone was able to disrupt the mature fibrils. But at the same time, they observed something. They realized that Juglone was also cytotoxic to normal cells. It could disrupt the mature fibrils in vitro, but when you introduce it into normal cells such as human epithelial kidney cells 293, they discovered that the Juglone was cytotoxic. So what's the relevance of these observations? The point is sometimes, please the next slide. The relevance here is sometimes we could observe a particular effect from an extract and then isolate the compounds and not have the same effect, because there are some things that also come into play when we use extract, which are not available when we use a purified compound, such as synergistic activity, such as additive effects, such as the ability of one compound to modulate the effect of other compounds. And at this point, if our interest is therapeutic efficacy, if we want to have the maximum therapeutic effect and drug effect, then we may not necessarily have to isolate a particular compound. We can do what has been a common knowledge in traditional medicine for centuries and millennia, which is we can enrich the extract to give us more of the fraction, more components in the fraction that is active to improve the therapeutic efficacy. So then, why is it important to isolate? Some people will ask. Yeah, there are benefits for isolation. It is through isolation that sometimes we identify the particular compound, because in some cases it is one compound that is producing most of the effect. Isolation gives us that. But with the advance in technology now that we have LCMS, we have MSMS, we have high resolution equipment to not just characterize and elucidate even the components in our extract, but also to sometimes even identify the particular group of compounds. In that case, we may not even have to isolate. But in the case where it is just one compound that is responsible for most of the bioactive effect, isolation gives us the advantage that that compound can now be produced using synthetic pathways. So we don't have to go back and start isolating it from natural sources. We can use synthetic pathways to isolate that compound. Isolation and structural elucidation also gives us avail us with the opportunity to discover new structural scaffolds that we can also use in drug development. That is important. But still on amyloidosis, I want to present a work that in a way contrasts what we observe in the case of Centella and in the case of Bacopa Moneri, which was in a recent work that was published in ACS Neuroscience on Rosa damascena. So this plant is common in the Middle East and also in Africa. We see it obviously as rose flowers. That's the way most people see it. And in the Middle East, it's used for occasions, it's used for spices, it's even used for food as spices, and it's used in perfumes. So the flowers of rose is rich in phenolic glycosides. So this research group wanted to see if the extract from those flowers, the methanolic extract from those flowers, had any effect on alpha-synuclein, which is the protein that is responsible for Parkinson's disease. When alpha-synuclein aggregates, it causes Parkinson's disease. So they wanted to see if the methanolic extract will have any effect. And yes, indeed, the methanolic extract from Rosa damascena, it did not prevent the aggregation of alpha-synuclein. But what it did was it bound to the alpha-synuclein and took it off the amyloidogenic pathway. It made the oligomers that were formed from alpha-synuclein to be less toxic than otherwise would have been. Because of that result which they observed, they decided to identify the metabolites that were present in that bioactive fraction. And they realized that the compounds there were gallic acid, camphorol glycoside, camphorol 3-O, beta-rutinoside, and quercetin. Now, I will stop there. You see, these compounds are compounds that even by the time I started my research, about 6 years ago, I already knew that these compounds had anti-amyloidogenic effects. You see? So you don't need to isolate these compounds. They are available in the market. You don't need to isolate them from ROS. But here is the relevance of this research. It's not particularly about the isolation. The relevance here is that they were able to establish that extracts from rose flowers could ameliorate or at least reduce Parkinson's disease, which is a devastating neuromuscular condition. And because rose is easily found in many parts of the world, even in poor countries, this gives us the opportunity of using this as a natural medicine, as an alternative. Not entirely, but we can use it to complement as an adjuvant, to supplement what we get from conventional treatment. So that was the relevance, that we can now deploy extracts from rose for combating Parkinson's disease at the population level. Please, next slide. So, after being able to establish that Centella asiatica had positive effects on transthyretin amyloidosis, at that point, I was also seeing some research showing that Centella asiatica had some beneficial effects on diabetes. I wanted to see if I was interested in whether Centella asiatica had the ability to inhibit the formation of advanced glycation end products. What are generally known as AGEs? What are AGEs? These are complex molecules which are produced in our body under physiological conditions, but they are accelerated during the production of, during hyperglycemic condition. They are accelerated, and they have the ability to accelerate the degeneration and complication that we often see in type 2 diabetes. So it has been recommended that one way of combating that we can help to reduce the symptoms and the morbidity in type 2 diabetes is by combating and preventing the formation of advanced glycation end product. So what I did in this research was I was able to prepare a phenolic-rich extract using a method that we recently developed in our lab. So by enriching the phenolics from Centella asiatica and using bovine serum albumin as our model of glycation, we discovered that in the presence of Centella isceltica, the formation of fluorescent advanced glycation products were drastically reduced. And the formation of protein oxidation products, such as dityrosine and N-formylkynurenine, were also substantially reduced. See, these products of protein oxidation are not just detrimental and implicated in the progression of diseases such as diabetes, obviously, but we also see them in other degenerative conditions such as in cancers. We also see them in cardiovascular diseases. We also see them in neurodegenerative diseases such as Alzheimer's and Parkinson. So the fact that the phenolic-rich extracts from Centella was able to reduce this, not just the advanced glycation products, but also the oxidation, the protein oxidation that comes with the formation of advanced glycation product. We realized that Centella extract could be relevant in reducing the effects of oxidative stress. And when we profile the Centella isceltica, we realized that it was basically the antioxidant and the anti-radical activities from Centella that was responsible for the anti-glycation and the anti-protein oxidation effects. So at this point, I want to round up, which is, today there's always, as a scientist in a discipline related to phytochemistry, we are always faced with the choice of whether to isolate after we have profiled for bioactivity. Here is what my little experience has shown me over the years. Sometimes it's relevant to isolate, especially if we are interested in identification of novel bioactive scaffolds, novel structures which we can use for synthetic chemistry. If we want to produce new drugs, if we want to have new drug candidates, sometimes we need to isolate. But if our interest is more related to therapeutic efficacy of the extract which we have profiled, then enrichment of the bioactive extract may be a better approach, especially given that today we have the tools to make that happen. We have supercritical free extraction, we have subcritical extraction methodologies, which can help to improve the bioactive component in an extract in remarkable levels. We have different extraction approach, green extraction approaches, which we can also employ to improve the bioactive component in any extract. And to top onto that, we also have the advanced tools which could help for the structural elucidation of the bioactive component without necessarily isolating the individual compounds. Today, we can use LCMS to identify the compounds that are present in our bioactive enriched extracts. And we can even quantify them. We can quantify the individual components, individual compounds in the extract using LCMS, using NMR. So Indeed, we are in a new era for the application of traditional medicine in combating neurodegenerative diseases. Last slide. So with that, I'll bring my talk to a conclusion. Thank you for having me. And I want to extend my deep appreciation to Dr. Yogesh for extending this invitation to me. Thanks to everyone who has joined us for this presentation. I'm grateful.
Thank you so much, Dr. Eze. I'm really glad to extend my vote of thanks to the Dr. Frederick Eze, who has given a session which was full of knowledge. And I'm sure the knowledge that he has shared in this session was will be a guiding path for the researcher in the future. And I request, before I conclude this session, I would like to request delegates, if they have any query, then please do ask. Please put it on chat box. We are continuously observing it. Don't hesitate to ask any query. I think no one has any query, as they have understood each and everything, whatever research work that we have done. And I'm sure that it will be a guiding path for their research that they will be doing in the future. And Dr. Frederick Ise, and thank you so much for taking out some time from your busy schedule. And you joined us for this second ICTM 2021. We are looking forward for your physical presence in third ICTM 2022. Thank you so much on behalf of SMBT Educational Trust and entire organizing committee of ICTM 2021. Thank you, Dr. Frederick. Thank you.
Thank you very much.
Thank you so much. We'll be having the next session sharp at 12:30 p.m. So please stay connected. Thank you so much.
साडेबारा झाले ना.
Good afternoon, everyone. Today I will introduce our third speaker in ICTM 2021, Dr. Slamet Videdo sir, MKS. Sir was completed the education in Master of Public Health Promotion and Medical Doctor Lecturer. Sir had many experience. Okay, in 2015, Sir worked as a Chief Committee of National Seminar on Hypnobirthing. In 2016, Sir has Chief Committee of National Seminar on Metabolic Syndrome. In 2017, the Chief Committee of National Seminar on Diabetes Update. In 2017, the Chief Committee of National Seminar Patient Safety Management. In 2018, the Chief Committee of National Seminar on Workshop on Complementary Alternative Medicines. In 2018, the Chief Committee of National Seminar on Patient Safety Management. In 2019, the Chief Committee of National Seminar Workshop on Tuberculosis Elimination. In 2019, Moderator of National Seminar on Triple Eliminations of HIV, Syphilis and Hepatitis. In 2019, Moderator of National Seminar on Advocating Paramedic, and in 2020, moderator of International Conference on Public Health International Meeting. Sir has organization experiences. In 2016, Sir worked as a member of Indonesia Mengajar. In 2010, Sir worked as a member of Palanmira, Indonesia. In 2015, Sir worked as a head of EDUCA operations, means education and health organization. And in 2018, Sir worked as a tuberculosis partnership program. So now I request you, Dr. Slamet Videdo sir, please continue our session.
Okay, thank you. Uh,
Sir,
Before I start my presentation, let me here to say hello to the head of organizing committee on the second international conference of on traditional medicine.
sir,
Mr. Yogesh will be here, and all the committee of the second international conference, and all the speakers, and let me here to bring my students of medical faculty of Malahayati University. Let me share my slide presentation. Is it okay? Is it okay for me to share my slide?
Yes, you can share your slide.
And actually, before I start my presentation, this research appropriate with the medical faculty maybe of SMBT because it really doesn't have correlation with traditional medicine, but the committee said that's okay to present on this nice occasion. Let me have the screen share, please. Is it okay for me to start my presentation? Hello, the moderator.
Yes, Professor, we can hear you. You can start your presentation.
Okay, thank you, Mr. Yogesh. Well, actually here I want to share about my research about obesity, and the title I give to my research is the analysis of risk factors for obesity occurs in the academic community of Malahayati University in 2020. Yeah, actually, I will not read this slide because the moderator has explained about my curriculum vitae. Okay, well, the audience and friends, speakers of this international conference, we know that obesity has been a serious problem in this world, not only in Indonesia, but around the world. Like World Health Organization said, actually it is not update, but in 2018 said that there has been 30% of the world population in this world being obese. So we can say that it has been a global epidemic. And how is the Indonesia condition of obesity itself? I take from the basic health research conducted by the Ministry of Health of Republic of Indonesia. The obesity incidence in Indonesia is even though it is not really high, but every year it has improvement. Start from the age over 15, the number is 30%. So every year to year, the incidence of obesity in Indonesia always increase. Relating to the obesity itself, my research will focus actually on the correlation between obesity and vitamin D controlled by the other factors such as gender and then socioeconomic factors, exercise or sport, heredity, food habits, sleeping and sleeping durations. Because those factors by the previous researchers have been researched. And actually, it is really interesting for me to see the relationship between obesity towards vitamin D. We know that the risk of obesity, like the World Health Organization explanation, obesity can cause some sickness or illness such as diabetes, hypertension, cancer, or maybe stroke. So from these phenomena, we concern on how to anticipate or how to decrease amount of obesity in Indonesia. It is the results of basic research conducted by the Ministry of Health of Republic Indonesia. Every year we can see here that in 2007, only 8.6%, but we can see here for overweight in 2018 become 13%. 13.6, while in obesity in 2007, it has been only 10.5%, but in 2018, it has been 20.8%. When we see the obesity number here, in 11 years old, it has increased more than 10%. So we should anticipate about this condition. Like my explanation before, the in this research, I want to see the relationship between obesity toward the vitamin D and other factors such as gender, socioeconomic, exercise, heredity, food habit, and sleep duration. Of course, in Malahayati University. And then we want to see the problem formulation from the case actually. Obesity and the other those factors, which one most correlated to the obesity itself? Vitamin D, gender, socioeconomic, exercise, heredity, food habit, or sleep durations. And later on we will see the condition. Well, for the purpose of this study, we want to see the relationship between vitamin D, that's serum 25 or HD, in the blood and the incidence of obesity. And the specific, we want to see the frequency distribution of gender, socioeconomic, exercise, heredity, and food habit, and then for sleep durations. And to study the relationship between gender, socioeconomic, heredity, food habit, and sleep duration to the incidence of obesity in the academic community of University of Malahayati. And the last, here we want to see the relationship between vitamin D levels in the blood to the incidence of obesity in academic community of University of Malahayati after control by gender, socioeconomic, exercise, heredity, food habit, and sleep durations as variable here. So talking about obesity, so what is obesity actually? Based on the World Health Organization, they say that obesity is kind of disorder involving exercise, body fat that can increase to the risk of health problems. And from the Sarif, it is the one of a pediatric doctor in Indonesia. He said that obesity can occur during being overweight compared to the ideal weight, which can be caused by accumulation of fat tissue. And the classification here, automatically, I differentiate with the WHO because based on the research by the Ministry of Health in Indonesia, we can say that someone get obese if their body mass index is more than 27. While in World Health Organization, they declare that for obese is more than 30 for obese. So the classification here determined by this formulation. And talking about vitamin D, so vitamin D is kind of a fat-soluble vitamin and has function which is needed by the body, and we can take it in our fat. And due to the nature of vitamin D, be able to dissolve in a fat, vitamin D can store and recover from our body. Because we want to see the risk factor of vitamin D deal with obesity, so we have the classification of vitamin D. If the vitamin D itself, I mean here also serum 25 OHD, more than 100 nanogram milliliter, it is potential toxic, and for the normal condition is 30 to 100 nanogram milliliter, and then for deficiency, if the serum 25 OHD is 10 to 29 nanogram milliliter, and we can see that if the condition is severe, in less than 10 nanogram milliliter. So the previous researches conducted by some researchers, obesity with vitamin D, by Van Lin, Simon Van Lin, I mean here, and the Hermawan. Hermawan is my advisor from Malahayati University also. And then obesity with gender, Cantor, and others. So it has been familiar actually, the research talking about obesity with its factor. So why I really interesting from this research, because based on the source and friend in 2011, she said that there is a correlation between vitamin D and obesity. The formulation we can explain here that when the vitamin D increase, so it will reduce the parathyroid hormones, and then the calcium will increase. Parathyroid decrease, calcium increase, and the effect of calcium increase will increase also the CSNS activity and then GAL tract. And for the parathyroid hormone decrease will influence the insulin sensitivity increase. Because of the activity of SNS, central nervous system here, increase, will increase thermogenesis, and then can decrease the de novo lipogenesis. Finally, the produce body fat. So from this theory, we believe that if the someone has vitamin D increase, can reduce the body fat. So with the short sentence, vitamin D high will decrease the obesity. So the conceptual framework I use here, vitamin D with lipogenesis, fat removal in phases, until the weight loss. Automatically, when we use this one, we have this design of research, my research, that we put vitamin D into independent variable, and the confounding variable here we have gender, socioeconomic, exercise regular, exercise, heredity, food habits, sleep duration. And we need the sample here, of course, the someone who obese and non-obese. And this research is a quantitative research. We use analytical, observational, and with the descent of case control. For the sample size, how many sample size we want? Because we need the serum 25 OHD here. We need the sample size to see the condition of vitamin D of our respondent. So by using sample size determination in health study with hypothesis test for population mean to site test, I take this one from the previous research by Parik. We have a number 28. At least participants or sample we can use here in one site is 28. So in this case, we use 30. We hope that it has been representative for my research. So we can see the characteristic of our respondent. We use 30 obese respondent and 30 non-obese respondent. But the results of the test, it's really a surprise for me. Why? Because for the normal condition of vitamin D, only one from the control group, and then for the case group, all the case group has deficiency of vitamin D. When we see to the formulation of this classification, wait a minute. Yeah, if you see the classification of deficiency from my research, we have from the case group 30 respondents and the control group 30 respondents, almost all of them have deficiency of vitamin D. And it is the characteristic of our respondents. And for male respondent, we have 27 respondents, and then for female, we have 33 respondents. Because it is, I think, a sample from the academic community, means it is not only student of medical faculty, but some my respondents could be doctor, lecturer, or staff of Malahayati University. Okay, from the results of my study, from an univariate analysis, we have that only one in normal or as a case group here all has a full deficiency, but only one in normal have normal. 29 have deficiency. And then for the gender, we can see here, not really different from the age, social economy, and then exercise, physical activity, heredity, food habit, and sleep duration also. So the discussion in this research is, even in the previous research said that there is correlation between vitamin D and obesity. Unfortunately, in my research, there is no correlation. Perhaps this, we have some trouble or some troubles in this research, and later on we are going to discuss it. So only one factor in confounding factor, that's heredity, has correlation with obesity. Even though the distribution of sample here doesn't have correlation, but as a researcher, I'm really interested. Why? Because almost respondent suffer deficiency of vitamin D. We know that from the theory, actually, usually for obese person has deficiency of vitamin D, but in my research, on normal person, they suffer deficiency vitamin D2. So the other factors, of course, happen to my respondents, not only about the obesity, but the other factor can cause the deficiency of vitamin D. Like the theory of this one, even my research is not appropriate with this theory, but I believe that in obesity, of course, there is correlation with the deficiency of vitamin D. So because I use this research for public health, so there are some discussion in public health that we want to try to connect with this condition. So we have like alternative solution maybe to cover this condition about the deficiency itself. There are four things like Suharman and Supardi said in 2018, how to avoid it. Illness behavior. And what is in illness behavior? actually, if the someone suffer deficiency of vitamin D, the someone itself should realize that take some capsule or tablet consists vitamin D, or by use sunbathing in the morning, because we know that the source of vitamin D come from food and the sunlight. And in this pandemic moment, we believe that the incidence of obesity will increase, because we know that we use physical distancing, and of course from this condition almost everyone will stay at home and automatically will avoid exercise, will avoid sport, and others will decrease many activities. It can cause the incidence of obesity be more increased. So the second solution maybe when we see here the health service behavior, taking vitamin D tablets or capsule is important if deficiency is known. Means if we have been a deficiency of vitamin D, so it doesn't mind that we should consume some tablet of vitamin D. And then for nutrition behavior, except the sunlight, vitamin D could be produced from our body by consuming some food consists of vitamin D, such as orange juice, egg, fish, shrimp, soybean, and many fish, tofu, maybe this traditional food from Indonesia, soybean cake, and mushroom, and many others food consists vitamin D. And then we can use the environmental health behavior. In this environmental health behavior, we should promote to many people that actually wearing umbrella, wearing clothes that cover the whole body, and other activities or habit that block our skin, it can cause the deficiency of vitamin D if this activity we always conduct every day. So by giving or by blocking sunlight to our skin or our body can reduce the composition of vitamin D to our body. Because this research even doesn't have correlation, I mean, for vitamin D and obesity. So we realize that, of course, there is a weakness of this research. So for the next research, maybe they should change the type of research. Maybe it is not case control anymore, but it can use like another approaches. And then the point is about the sample of size, because when I see the previous research of vitamin D, most of those researchers using more than 300 sample size, but in this research only use 60 if we back to the previous sample size, 30 obese person and 30 non-obese person. So when we use the statistical analysis, it is really small, but yes, that's okay for me because we have the limitation of time to do this research. And we think that this research has weakness on amount of sample we have. So the correlation cannot be proven. And then this is the situation we have when we conduct this research, because it has clinical research. We have a collaboration with Prodia as the institution who can take this serum 25 OHD automatically with Malahayati University. And how to determine about the obesity? Of course, we use BMI or body mass index. We use the body mass index. We account the body mass index from the height or tall and the weight. The weight divided into the tall in meter square. So if the BMI is more than 27, so we put into obese persons. But if the BMI is less than 27, so we say that it is normal or non-obese. So by having weight measurement and height measurement, we can see the body mass index of our sample. And actually, to see the obesity is not only about BMI, but maybe we can use the belly measurement. But we use in this research is BMI. And of course, we use the questionnaire itself for to see the condition of their sleep duration, their exercise, their food habit, their heredity, and the other factor we use for our research here. Okay, I think that's all before this one. So because I'm from Malahayati, so I want to introduce to you all about Malahayati. We are the one of private university in Lampung. We have medical faculty, public health faculty, engineering faculty, economic, and also law faculty. In medical faculty, we have nurse, we have medical doctor, pharmacy, nursing, and midwifery. So after pandemic finish, maybe we can conduct an international conference in Malahayati or in SMBT. We can have known each other to elaborate our research. It is the picture of my hospital, of Malahayati Hospital, Education Hospital, and then for the campus. And this one is library of Malahayati. We have the interesting library because we use all ethnic in Indonesia become our miniature of our library. And then because we have dormitory, so all medical student of Malahayati University stay in the dorm. So we have the dormitory. It is the picture of our dormitory. And then in Lampung also, you can see if you come to Lampung, we have many tourism places such as Waikambas. It is National Park of 11 in Lampung, and then we have Purbakala site. It is prehistoric site. It is in Lampung also, and we have many beaches. So like Pahawang, Kiloan, yeah, interesting beaches, and of course, when we want to visit Lampung, we can see these places. Okay, I think that's all for my presentation about obesity and some factors influence. Thank you.
Thank you so much, Professor Slamet. Now I request delegates, participants, if you have any query regarding particular topic, share with us.
One question from my side, Sir Widodo. Yes, I'm on to you. Yes, Professor. I am
Yes,
audible
Mr.
to you.
Yaki.
Which will you share about the latest data of the obesity in Indonesia?
Yeah, the data itself actually from the research conducted by the Ministry of Health of Republic of Indonesia. And actually, even it is not really high compared with India, but I think it is become like awareness for us to anticipate the obesity happen in Indonesia. Because when we see year to year, the number always increase.
Okay. So that's much from my side only. Okay. Thank you, Professor.
Okay, you're welcome, Mr. Yes.
Okay, any participants who are willing to ask any question? Yeah, I think no one have any question. Professor, thank you so much for giving us your precious time. You took out some time. I think we have one query. Do you think your findings in academic community of the University of Malahayati will be same if it was to be conducted in Africa? So this was the question from one of the delegate we are having from Nigeria, Dr. Chinedu Emu. He is asking question.
So the population here is in the academic community, like my explanation before that even yeah we have the same condition maybe. So we think it is the weakness of my research also maybe because almost a student in Malahayati stay in the dorm. We know when they stay in the dorm, they usually they don't have more exercise outside. And then for the lecturer, doctor, or maybe nurse here, I take as my sample. Also, relative we have same activity. So not really various. It also become the weakness of this research, I think, because
I think there is a slight technical problem. Wait for some time so that Professor Vidu will continue with his answer.
चालू आहे सेशन चालू आहे आजचा तिसरा चालू आहे ते बरे इंटरनॅशनल डेलिगेट जे
I think due to some technical problem, Professor Vidudu has disconnected from this session. But I would like to extend my thanks to Professor Slamet Vidudu, who has given his precious time from his busy schedule and accepted our invitation for the second International Conference on Traditional Medicine. I would like to thank all the participants and the delegates
yeah
who
yeah the sorry to interrupt you Professor Suryavanshi, there is a might a question into the your chat section. Okay, and you try to connect again to Professor Vidodo.
yes, sir. We
Yes.
are connected.
Yes,
Okay.
the Professor Widodo is with us again. Please, please.
The next question is, what do you advise academics to do so as not to be obese, considering the nature of the profession, which includes sitting for hours to write papers? Really valid
Okay.
for all of us,
Yeah,
for academicians.
like my profession at this moment becoming health promoter, how to avoid obesity. Of course, we realize that most of the incident of obesity because of the food consumption. So we should decrease the food consumption. We should select the food we want to eat, such as low fat, low carbo, and others. And we should do more exercise to burn the fat and carbo in our body. And of course, by applying healthy life, by consuming vegetables, fruit, it can decrease or it can eliminate the risk of obesity. Actually, even it is not really a serious sickness or illness, but we know that obesity can cause serious illness such as hypertension, stroke, and many cancer, colon cancer, and others. So if we suffer obesity, it is really dangerous for us to have those sickness. And this pandemic, COVID-19, based on the research at this moment, obesity will be one of comorbid in COVID-19. So it is really interesting when we want to do the research about obesity and the risk of COVID-19. So I think this topic still being interesting topic for the future.
Thank you so much. I think the explanation is satisfactory for the questioner who asked this question. So, is there anyone who would like to ask any query or question? Yeah, I think that's it for the delegates. And now, thank you so much, Professor Samet. After
Yeah, actually, I bring many my student here. Those are my medical student in the fifth semester, and then my nursing student also. Because it is the first time maybe for them to join international conference via online, and I really thanks to SMBT that has conducted this conference. We hope that for the next year we can conduct in the real meeting, the real situation. We can have a meeting. We can see directly. So maybe in Indonesia or maybe in India, because we have made a memorandum of understanding between SMBT Sabavati and University of Malahayati.
sure, Professor. Sure, Professor. We'll meet physically, but we are expecting, before that, we are expecting your physical presence in third international conference on traditional medicine, which will be held in 2022.
Yeah, maybe the last year, maybe we need like input from this conference because we are not really common with traditional medicine. So maybe for the next conference, we want to try to conduct in a general issue that's in medical in general. So maybe our doctor, our nurse, and midwifery will join in this conference.
Definitely. Okay. Thank you so much, Professor Raman Vidudo, for giving us your precious time. Thank you so much.
Thank you, sir.
Thank you, Mr. Yogesh.
is one announcement to all the participants. We'll be starting with the presentation session, scientific presentation session at 3 o'clock. The concerned Zoom links have been sent to you. Please connect to that link. Thank you so much. We'll see at 3 p.m. Thank you so much.